So for the final talk in this session, it's a great privilege to introduce Lucy, who's known to everyone, I hope, from the Royal Vet College, who's going to talk to us about susceptibility to coronavirus outcomes in relation to FIP.
Okay, it's the hardcore feline people left at the end of the day. I realise it's been a long day and I'm standing between you and a drinks reception, so I'm not going to dwell on too many things. I'm very happy to talk to people later. I've also put my email address there for people to email me if they have any questions. I'd like to return us now to the whole idea of, are there some cats who are destined to get FIP, and others, it doesn't matter what virus we throw at them, they will not get FIP. So it's the idea of there being a host genetic susceptibility here that my group are very keen to explore. I should preface by the fact that I am actually an internist. I'm a small animal medicine person. I'm actually an endocrinologist, so I've got no real reason to be here. But what happened during the pandemic, I was working on multi-species diabetes genetics, essentially. And of course, in lockdown, we were not permitted to go and work in the lab because nobody thought that canine diabetes was of national importance, which is fair enough. But actually, my lab is in Oxford, although I work clinically at the RVC and surrounded by many people putting a huge amount of effort into lots of things to do with COVID. I kind of had a bit of FOMO and I wanted to get back in the lab with the group. And we were fortunate that UKRI agreed with us that it was going to be worth us thinking about exploring companion animal coronaviruses. And I was able to assemble a group of colleagues who were keen to participate in what we ended up calling MASCOT, to look at animal susceptibility to coronaviruses from a host genetic perspective, and also to look at some transcriptomics. You'll see there's a dog and a cat in our logo. I'm focusing purely on our cat work, but pretty much what we're doing in FIP, we're actually also doing in dogs for canine respiratory coronavirus as well, but I'm not going to talk about that today. I want to share my acknowledgements first, because I realise people will melt away towards the end. There's a huge number of people who've been involved with this, and this would not have been possible without all of the people listed here and many others. The four people to whom most of this data is owed today, Dr. Emmy Barker and the team at Bristol, Severine Tasker, and all of those over the years who've assembled that database and archive of samples, Marcia and Tom, who have done a lot of the bioinformatics work, and Sarah Falcone, who's done an awful lot of the hands-on sample processing. But there are many others involved in the MASCOT group. We met very frequently to begin with. We now meet still very regularly, but less frequently. And we're fortunate to have perspectives with human medicine, genomics, virus genomics, and veterinary medicine, as well as pathology and a range of other experts on the group. I also want to acknowledge the other people who've provided us with samples, expertise, or phenotypic data on the samples that we've been using. Particularly, Anya's done all the diagnostics on the Bristol archive. In terms of immunohistochemistry. We've got a great collaboration with APHA. We've been looking at some of their SARS-CoV-2 infected cat samples. And in terms of feline genomics, so host genomics in cats, actually the famous person in that field is Leslie Lyons, who runs the 99 Lives Consortium, and it's really the only shared database of feline host whole genome sequences, and she's been extremely generous as well, sharing data and expertise with us. The good thing about going towards the end, I don't really need to talk about where the feline coronavirus sits, and I don't really need to tell anyone in this audience how mysterious it is. We know from a host genetics perspective that there are factors that make a cat more likely to develop FIP should it meet the feline coronavirus. We know that certain breeds are predisposed. We know that age also plays a role, but we don't really know why. And there are other aspects of feline infectious peritonitis that have come to the fore from things like transcriptomic studies. We've learned an awful lot from the recent outbreak in Cyprus, but we still can't tell, looking at a cat, what's going to happen if it meets the coronavirus. We just don't have the capacity to do that. And again, I'm going to go through this quite quickly because this has been talked about quite a lot today, but we do think that the process of meeting the virus and what happens is likely to have a host component to it. Some cats are resistant, some cats carry on shedding, and some cats persistently shed into the long term, and we do think there may be a host genetic component to that, as well as a viral component. And we know that we've talked a lot today about the various things that must happen or may happen to contribute to a cat developing FIP, and it's clear that that's quite a mixed bag of things. I don't think all of them are necessary. I don't think any single thing is sufficient. I think we're looking at a complicated, multifactorial process. So the reason this arose is, of course, working in Oxford, I'm surrounded by people who are obsessed with genotype-phenotype correlation in humans and understanding immunogenetics, both in health and what our phenotype is like at rest versus what happens when we develop a disease. And there are many, many publications about host susceptibility in COVID-19, various different polymorphisms that have been described that impact on whether a person is going to make a quick recovery, whether they're going to have severe symptoms. And there's all kinds of really fascinating evolutionary history that's come through from those studies as well. When we look at cats, slightly more patchy data out there. We can go back to this paper in 2006, and what we see is that certain breeds are predisposed to FIP, and that does indicate there is a susceptibility within those breeds. But of course, within some of the more exotic purebred cats, the husbandry of those cats is also different. They're often bred within breeder catteries, and so they have a different experience at the beginning of life, perhaps, to some of the stray cats that we see. But definitely, we know that we have an overrepresentation of purebred cats within our FIP population. Having said that, coming through the clinic at the RVC, most of the cats we see with FIP are domestic shorthairs. So in a numbers game, it really is the domestic shorthairs that are giving us the highest number of cats. And I'm sure everyone in the audience is very well aware of how host genetic variants can contribute to risk in a complex disease. But I just want to run through the three categories that we sort of have tried to classify these into, really learning from our experience with looking at diabetes in dogs, actually, which, as I've talked to people about today, in some ways, diabetes and FIP are not that far apart, because there is a host susceptibility or a baseline susceptibility in the individual, but there are also fairly stochastic random events in the environment that also contribute. Now, we know a lot more about those events in FIP because we know there's a virus. We're not so familiar with all the various triggers in diabetes, but we have to model that uncertainty when we're trying to understand the genetic susceptibility, and we have to be a little bit more flexible in our definition of control in those studies, because an animal may be a control simply because it hasn't met the right environmental trigger at the right time. It may yet carry the genetic burden that would have given it the disease had it met the right trigger. So we've tried to really classify the variants that we're interested in into three categories. There are always going to be variants that are associated with a particular breed. So they're going to be high frequency in a high risk breed, and they're likely to be quite fixed in that breed because it's been squeezed through a genetic bottleneck to make the breed. And that might make the whole breed, say the Burman cat, predisposed to FIP. Now, if you look at cats with and without FIP in the Burman breed, you might not see any difference. They might all carry that susceptibility. The only difference is who has met the virus. There are also variants within the breed where you are going to modify susceptibility within that breed. So you're looking at cases and controls within a single breed. And those would be variants that are higher frequency in, for example, those Burman cats who do have FIP versus those who don't. And then there are always unusual, usually very high impact, strange cytokine polymorphisms, things like this, that really do have a dramatic effect in individuals on their predisposition to disease, and that can teach us a lot about the disease, but it's not something we would look for in this type of study. And we recognise that FIP is likely to be a complex polygenic trait. This has been said before in other contexts today. We're again all about association. We're not really looking necessarily primarily at causality, although we do have now some work in progress trying to look at the function of some of these variants. And I'm sure those of you who work with genomic data are very familiar with single nucleotide polymorphisms, the most common type of variation in the feline genome, but we've also got other things, insertions, deletions, all kinds of variants that are structural. And some SNPs are non-synonymous and they will change the amino acids. Some don't change the amino acids, but we know, particularly from looking at COVID and looking at other complex traits in humans, even SNPs that don't change the amino acid can be really important in gene function, whether that's through changing the stability of the gene when it's transcribed or changing the way it's spliced. We're slightly more excited by synonymous SNPs than maybe we used to be a few years ago, because we really know that they have quite often a regulatory function. Even genes in introns and in gene deserts, where there are no genes at all, even variants in those locations can be important in the way in which the chromatin is accessed, for example, other regulatory functions like transcription factor binding. Anywhere in the genome we're interested in, but we do still favour those variants that change amino acid sequence, because it's kind of easier to convince people that there's a biological reason that they're important in risk. So I'm not going to dwell on our RNA sequencing data, other than to say that we have done some RNA-seq of different tissues in cats with FIP from the Bristol archive. What I really wanted to show, essentially, in this slide, I'm going to see if I can use a. Let's see if I can use a laser pointer here. These are some control and FIP cats. Each vertical column is a different cat. I think not only does this show that there are differences between control and FIP cats according to the whether they have looking at the different tissues, what we can also see is that there's a lot of variation across different cats, even with the same disease. So we can see that some cats really show a very striking change in the transcriptome. Some cats, it's a pretty quiescent situation. But overall, we know that when we look at tissues affected by FIP, it's pretty much all about the type 1 interferon response. That seems to be the genes that are differentially expressed. So what about host genetics? Well, previous work has really been based on what we already know about the cat genome, which actually is not very much. So we know some of the variation from a single feline reference, which happens to be an Abyssinian cat. And we know we have one SNP chip in cats, but it only has about 60,000 variants on it. And we've got an idea that there may be some candidate genes that could be important. So that's what previous work focused on. And we've seen published previously, before we started this work, SNPs in genes associated with the immune system, like interferon gamma, TNF-alpha polymorphisms. We've seen other variants associated with coronavirus shedding. And there has been a genome-wide association study done in the Burman cat. And that did indicate within that breed, so these are those class 2 or type 2 associations within a breed looking at cases and controls. In the Burman, there were 5 regions that were associated with risk of FIP, but none of them were completely concordant with the disease phenotype. So as I said, a lot of the work's been done with SNP chips, and it's been done with candidate gene studies. And the SNP chips can be extremely helpful if they're very densely packed and we have good knowledge of the genome that we're studying. We can look at tens of thousands of variants, and they help us to focus in on a particular region of the genome, but we're missing a lot of information from SNP chips. So we decided to use whole genome sequencing of the cats this time, rather than the viruses. And this gives us a more comprehensive evaluation of millions of variants within each patient. And it gives us the potential to discover novel variants. We can annotate the variants about whether we think they're going to be high impact or not. We've got to bear in mind it's not going to give us epigenetic information or structural variants, but it still will give us a broader perspective on FIP susceptibility. So this is the feline arm of the first MASCOT grant that we had, and we just took 12 cats from the Bristol archive that had FIP and 12 cats that definitely did not have FIP that were older. And we've actually also, as well as doing whole genome sequencing on those cats, because we were interested in the feline MHC, and that is incredibly poorly covered in the current feline assembly, we decided to make our own feline nanopore assembly to try and see if we could pin down the MHC region a bit better. And we fed into this pipeline the data from the RNA sequencing from the same cats, actually. And what we've done, because this is not a big study, it's not like the studies you see in COVID-19 where the NIHR and the NHS and all of these different organizations have got together. This is small. We recognize the power is not massive. So the way that we've tried to address that is to take our whole genome sequencing data and validate it with targeted sequencing in a bigger cohort, and then take the data from that and do another round of validation in a completely different cohort. So we've tried each time to iterate our results and get closer to the actual variants that are associated with disease in the 3 separate processes, because we really hope, what we told the UKRI as well, was that this is going to help us identify novel factors for susceptibility to coronavirus-related diseases. It will help us understand companion animal coronaviruses and the response, knowing that there's the potential for zoonosis in future. And so Tom really led on the nanopore and the RNA-seq, and Marsha has done the whole genome sequencing and the follow-up. Now, one of the challenges with using whole genome sequencing and small numbers to try to identify associated variants is you end up with millions of variants, and then you somehow have to filter them to work out which you really think are plausibly, realistically likely to be associated with your disease of interest. And anyone who wants to talk about the bioinformatics, I'm happy to do that. I'm not going to go into a lot of detail today, but essentially we take the whole genomes, follow a traditional GA-TK pathway. But what we've developed over the years, and this has sort of been stolen from our canine diabetes work and adapted for FIP, is a variant prioritization pathway where we take data from as many sources as we can, not just to do with a species of interest, but a lot of human data as well. And we pull all that information together and we decide roughly what weight we want to give each piece of evidence, and we come up with an evidence score for that particular variant so that we really prioritize looking at the variants with the highest evidence score, and we do our follow-up genotyping on those variants. So how does that work? Well, in reality, it's a bit of a moving target, and it requires a lot of discussion. It's a bit like standing in front of a big machine and trying to get a very small number of jelly beans to come out of the bottom by turning various dials. You just really want to focus in on those variants that you think are the most important. We're going to miss things because we're already bringing the numbers down into quite small numbers of variants, but we are hopefully going to identify those that are really critical. So we look at the frequency of the allele, obviously, in the cases versus the control cats. And we look in other databases, as I mentioned, the 99 Lives database of cats, mostly US-based cats, and we see how common this allele is in the population. We think about the predicted impact of the allele. Is it high impact? If it is, we might prioritize it a little bit more highly. Is it in or near a gene that could be relevant plausibly to FIP? So is it near a gene that's been involved in COVID or viral infection or in another study? And is it differentially expressed in our RNA-seq data? And there's loads of resources we can use for this. I won't go into all of them, but it's makes you excited, but also a little bit depressed to see how much is out there for human data and how little is out there for companion animal species. But we've been fortunate to be able to access open access work, as well as Professor Leslie Lyons's 341 domestic short-hair cats and other breeds. And actually, one of the things that Leslie did was extremely kind and very valuable to us. We knew there were some FIP cats in that data set, and she very kindly told us which they were, so we were able to make sure that the cats that we looked at didn't have FIP. Ooh. Okay, so I've talked about FIP, I've talked about our whole genome sequencing, and then I've talked about the way in which host genetics can contribute to the disease. And I would say that at the moment, what I'm going to show you is really mostly related to those class 2 variants in domestic shorthair, but we do have information on other breeds as well. And from the whole genome sequencing, part 1 of the study, we found 38.6 million variants. This is why we need bioinformatics and a filtration program. But I think the key point here is that when you look at those variants, only 2.5 million of them had been previously published in cats. So 36 million were never going to be in a candidate gene study or on a SNP chip because they hadn't yet been published. So this is why we feel that there's more novelty in this study by doing the whole genome sequencing, because we were able to take all of these variants, make it sound simple, but we applied our evidence-based scoring system, and then we took the top variants with the highest evidence score to another cohort of cats and did follow-up genotyping. We actually took 10,000 variants from that study, and we took 66 more FIP cases, 39 controls, and we analysed those to see whether the association persisted in a second round, and we did some MHC sequencing, which I'll talk about very briefly at the end. Now, most of you will be familiar with these Manhattan plots, which show you the whole genome across the x-axis, and then on the y-axis you have a value that's essentially a statistical measure of how likely that individual dot, which is a SNP, is associated with the phenotype of interest here, FIP. And we recognise this is not a massively powered study because it's still very small, so we have to be a little bit generous with our p-values. But what you can see is you're starting to see some areas of the genome where there are some SNPs that rise above the others from our whole genome sequencing and our phase 2 genotyping. Let me just show you a couple of examples to kind of zoom in from that multi-million SNP study into 3 examples of variants that would have been of interest to us in that phase 2 to take forward into phase 3. So these are stacked bar plots, and they represent 3 different regions of the feline genome, 3 different SNPs. And where the bar is red, then we have the cat is homozygous for an allele that is not the published reference allele. Where it's blue, the cats are heterozygous, and where it's green, the cats are homozygous for the reference allele. And what you can see on the different columns of the stacked bar plot, we've got our domestic short-hair control cats and our domestic short-hair FIP cats. And then we've got our full cohort, which includes not just domestic short-hair, but all the other breeds, control and FIP cats. So in this first one here, you can see that the majority of the control cats are homozygous for the reference, but you can see amongst the FIP cats in the domestic short-hair and the full cohort, there are more heterozygotes, and actually all of the homozygous alternate allele cats have FIP. Something similar in the second variant, and in the third one it's inverted. It's actually the homozygous alternate allele that seems to be protective. Now we've got millions of these, we've got, well, tens of thousands of these, but actually what makes these interesting is we start to look at what the variants are. So this is a missense variant in a gene with antiviral activity. It's associated with COVID-19 risk and hospitalization. This is an immune sensor involved in the immune response. It's only expressed in monocytes and macrophages, and it's a diagnostic parameter for macrophage activation. And this is another immune receptor. So these things start to look quite exciting, and after the second round of genotyping, this is what we had, a number of genes, a number of variants in which we saw multiple lines of evidence for their association with that cat being more likely to get FIP, ranging from whether it was a COVID-19 gene or it'd been published in FIP before, or it's an immune gene, all kinds of things to take us closer to publication. But we know the numbers are small here, and what you see in our odds ratios are massive confidence intervals, and we really wanted to try and shrink those down. So that's why we actually then went to the RVC clinical records and archive, and we pulled out all the RVC FIP cats. So we did phase 3. And actually we were fortunate here. Marsha and I applied for some money to the UK ICN, and we got some funding to support targeted sequencing of our favourite 3,000. Now we'd narrowed it down to, in the RVC, cases and controls, 89 cases, 115 controls. And essentially all the data here has now been examined in a meta-analysis, and this has not been published, and I'm sorry I have to be a little bit circumspect in the genes here because we can't release them just yet. But what we have essentially are 23 variants that are associated with FIP risk. In the domestic short hair, you can see our odds ratios, our confidence intervals have tightened. There are 13 variants here, 9 of which are associated with increased risk, 4 of which are associated with protection. And these are odds ratios in the region of making a cat 2 or 3 times more likely to develop the disease. We also have a similar set of data from the full cohort, including many other breeds of cat. And actually, if we combine all this data, we actually see that 3 of those variants overlap in the 2 cohorts, so we have 23 new variants in total. And what I will say, I can't tell you the exact genes, I'm sorry, but what I will say is the word macrophage really strongly pops up here. This, to me, is a macrophage gene set. Many of these are actually intronic. Yeah, I know somebody's excited in the corner. But what I would say is that many of these are actually non-coding. They're intronic. They're in places that are close to macrophage genes, or they're in them, but they're not. There actually isn't anything that changes the coding sequence. But I do think it gives us a really strong indication that it's a cat's macrophage behavior that is doing something to the way in which it interacts with the virus. The other thing that's really strongly on my mind based on my diabetes background is the fact that whole genome sequencing doesn't capture things like MHC and other sort of structural variants as well. And we know that MHC is also associated with SARS-CoV-2, and of course we know that MHC is associated with risk and outcomes in other species with coronavirus. And MHC, of course, it's likely to be involved in your immune repertoire, in the way in which you interact with an infectious disease, what antigens get presented. So whilst we've got this story coming from macrophages, whether that's as part of the innate immune system or as antigen-presenting cells, we've also got this idea that maybe the MHC is important. And the trouble with the feline MHC is that actually nobody's really had time or the resources to look at it properly. So we thought we would try and make a bit of a contribution here, and we used some of our funding to make a new assembly of the feline genome from an FIP cat. And so what you can see here on the left is the old Yuki et al. Assembly, and this gives you an idea of how contiguous the assembly is. The blue bit here, the blue vertical stripes show you where the MHC class 2 is. The pink bits show you where the MHC class 1 is. And as you can see, it hasn't really been assembled before. So we've built a new assembly. This has been done by Tom, and we have managed to assemble that region now with the help of Matt Luce at Nottingham and Lorna Kennedy, who is the international expert on canine feline MHC. And this, again, is work in progress. It's extraordinarily expensive to do this work because we have to use a technology called read-until just to sequence that part of the feline genome which contains the MHC. And it has been quite a time-consuming, expensive business. But we've done 26 cats now, including 10 with FIP. And what you can see here, I know this is quite a complex slide, but there are 3 different loci here, the DRB1 locus, DRB3, and DRB4. These are MHC class 2 loci in cats. And they're very polymorphic, so lots of different alleles that could be there. And all these different potential alleles at this locus are shown across the bottom here. And what we've got in blue is the FIP cats, and in red is the control cats. And you can see we've sort of tried to separate out the 26 cats according to their genotype across the different alleles. There isn't a single MHC haplotype that is the FIP haplotype. I don't think any of us were really expecting that. However, what we can see are there are a couple of candidate regions where if you've got that allele, then that's an indicator you're likely to be in the FIP group. There's one here in the DRB1 locus, and there's one here in the DRB3 locus. So we are excited about that, but trying to find a cost-effective way to look at more cats. So I'm recognizing that I'm running out of time, but just what we're doing at the moment, we are applying some of our genetic work to cats who have seroconverted to SARS-CoV-2 in collaboration with Margaret Hosey and her team in Glasgow. We're doing functional work on our associated variants. We've been pulling out viral sequences from our RNA-seq, and we've also got some interesting data on immune repertoire as well on our affected and unaffected cats that is starting to show us some quite interesting information about what happens to the cat's immune repertoire after they've met a coronavirus, whether they have FIP or not. We're doing some annotation of the feline genome to try to improve our knowledge of regulatory elements, and we are starting to look at class 1 variants, response to treatment, and we're really keen to collaborate with others. So is it likely we're going to be able to do a polygenic risk score and have a crystal ball to tell you who is going to get FIP? Maybe, not with this data at the size it is, but I think we could probably gather the cats together who we think are more likely. Because this is such a multifactorial problem, we're going to need more cats, more countries, more collaborations, because there are so many other things there that are involved in the risk. So I hope that I've showed you that you can use whole genome sequencing of the host for complex diseases, and that if you have the capacity to pull in data from other people's studies, that can also help to inform the work that you're doing. And I just want to say again, thank you for the invitation. It's been amazing to be here and have a whole day of feline coronaviruses. It's a real treat. And thank you to those people who are not here but have really done the lion's share of the work in this. And I'm really happy to answer any questions. Thank you.
Amazing. Thanks very much, Lucy. So time for a couple of questions before we break for drinks.
It will just be a quick one. When you were looking at the FLA and you saw that there was a couple that could be associated with the disease, is there the opposite, where there's some that could be protective against?
It's very hard to know. I think at the moment we have focused on risk rather than protection, because we don't really have a sense of what the diversity is out there in the general cat population. I think that will come, and there probably will be some protective alleles. And I think that's going to be important also in knowing, for example, if you're going to intervene with a vaccine or something like that, you know, who's got natural protection and who hasn't. But I think we're not powered for that at the moment. We're really just looking to see what was shared amongst the cases. I should also say that as we all piggyback data in studies, you can imagine that some of the other cats in the studies had other diseases that weren't FIP, so that could also influence what haplotypes they had too.
Thank you so much for your presentation. I was wondering, with the polymorphisms that you were looking at, I know you can't name the genes, but have you noticed, or is there any current research on the heritability of those particular genes?
It's a good question. We literally, what I showed you, that's why it was a bit grainy. It's pretty hot off the press. What we have done in terms of looking in other populations has really just been done with 99 lives. What we know is that of the variants that we're associating with FIP, they are at relatively low frequency in the general cat 99 lives cohort, but in terms of heritability, I would expect it just to be normal, standard Mendelian inheritance, but we don't know. That's the bottom line. We haven't really had a chance to explore that in pedigrees or anything like that. But you're right, you're thinking about whether or not there might be a lethal effect or something like that?
Actually, I was wondering if there was maybe some sort of potential for breeding.
Yeah, breeding away from...
Yes.
I think it's the same with diabetes. Actually, you're much more likely to be able to control the environmental trigger if you can find it. And then you have to focus your protective efforts on those who are exquisitely susceptible. I think we are probably never going to be able to breed risk out. And if we do that, there's a risk we'll breed something else in. I think it's unlikely there'll be a single genetic test for susceptibility of FIP in cats, unless we find something in our breed unique stuff that we've started to do.
Okay, thank you very much.
Thanks.
Okay.
Maybe it's a sign that we don't need to speak.
I'm fascinated by this 99 cats thing. Not quite enough to donate yet, but how is it going to? I just sort of, is there the opportunity to use that data in a sort of a pan-genome type analysis, so you can map to all variants rather than just a single reference? Or is it quite a controlled data set?
It's quite a controlled data set, and I think the other thing is that most people doing the work are donating. We've donated our genomes to it now, so it becomes a bit self-fulfilling. Yes, there is an effort to make a feline pan-genome that represents all the variability in cats. And what we have done, and why we've been so keen to get UK cats into it, is that it's also informed the design of subsequent SNP chips and things like that, because they haven't been well covered. So the way it's released specifically is everybody gets a VCF once a year containing all the variants.
And UKRI are fine with funding that sort of data access, do you know? Sorry, that's a bit of a...
We told them that's the only way we can share it and make a difference. You know, I mean, there's 300 odd cats in it now, and we've probably donated, I don't know, 12. We've kept the cases back until we're published. We've given them the 12 controls, and we've given them some other cats of ours.
There's nothing to stop sharing your data more widely.
No, there isn't, because we don't share the clinical phenotype data. We just say, this is RVC Cat 1. And if there is a study that really goes, oh, we found something weird in one of your cats, it goes back to 99 Lives, it comes back to us, and we have discretion over whether we choose to release the phenotypic information on that cat. So it's basically just a cat genome, because there are so few out there.
Okay.
We could do with a much better model, but actually, the UKRI don't appear to be that interested in cat genomics.
Well, I can tell you, last time I looked at it, I decided that it could sit on my lap if it wanted to. I wasn't going to force it to do anything that I didn't want it to do. They didn't want it to.
Pretty much.
Thank you.
Okay, in which case, can we thank Lucy and all the speakers for a great set of talks today.